Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs, divided animals into two groups that had red blood and that did not. In Linnaeus' time, a two-kingdom system of classification with plant and animal kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic, that is green algae, and non-photosynthetic, that is fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, two-kingdom system of classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Linnaeus gave two-kingdom classification. Haeckel gave three-kingdom classification. He included Protista. Copeland gave four-kingdom classification, included Monera. Carl Woese gave six-kingdom classification with three-domain systems, Archaea, and R.H. Whittaker in 1969 gave five-kingdom system of classification. Characteristics of five kingdoms. Monera, Protista, fungi, plantae, and animalia. Monera, most abundant microorganisms, unicellular, cell type prokaryotic, cell wall non-cellulosic, polysaccharide and amino acids, made of peptidoglycan. Nuclear membrane is absent, body organization cellular, mode of nutrition autotrophic, that is chemosynthetic and photosynthetic both, and also heterotrophic, saprophytic, parasitic, shows highest nutritional diversity. Protista, unicellular, cell type eukaryotic, cell wall present in some, nuclear membrane present, body organization cellular, mode of nutrition autotrophic, photosynthetic, and heterotrophic. Fungi, eukaryotic, present cell wall is present without cellulose, are made of chitin. Nuclear membrane present, body organization is multicellular or loose tissue. Mode of nutrition heterotrophic, saprophytic, parasitic. They are cosmopolitan, occur everywhere. Plantae, eukaryotic, cell wall is cellulosic, nuclear membrane is present, body organization tissue or organ level, autotrophic, that is photosynthetic. Animalia, eukaryotic, cell wall is absent, nuclear membrane is present, body organization is tissue, organ or organ system level, heterotrophic, holozoic or saprophytic. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were Monera, Protista, fungi, plantae, and animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a six-kingdom classification by Carl Woese. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. These placed together groups which widely differed in other characteristics. It brought together prokaryotic bacteria and blue-green algae, that is cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. Fungi had chitinous, while green plants had cellulosic cell wall. When such characteristics were considered, fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped under kingdom Monera, and unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants, and both having cell walls, with Parametrium and amoeba, which were earlier placed in animal kingdom which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only morphological, physiological, and reproductive similarities, but it is also phylogenetic, that is based on evolutionary relationships. Kingdom Monera, divided into archaebacteria and eubacteria. Bacteria are the sole members of Kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus cocci, the rod-shaped bacillus bacilli, comma-shaped vibrium vibrio, and the spiral spirilla. Bacteria of different shapes: cocci spherical, bacilli rod-shaped, it has spores, spirilla flagellum spiral, and vibrio comma shaped. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria have special since they live in some of the most harsh habitats, such as extreme salt areas, halophils, hot springs, thermoacidophils, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, responsible for production of methane, that is biogas, from the dung of these animals. Eubacteria. True bacteria. If flagella is present, made of flagellin proteins. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum made of flagellin protein. The cyanobacteria, that is blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocyst, example nostoc and anibina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Most of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Diagram of a filamentous blue-green algae, nostoc. Heterocyst, mucilaginous sheath or gelatinous sheath. Some heterotrophs are pathogens causing damage to human beings, crop, farm animals, and pets. Chlorella typhoid, tetanus, or lockjaw disease, citrus canker are well-known diseases caused by different bacteria. Cholera by Vibrio cholerae, typhoid by Salmonella typhi, tetanus by Clostridium tetani, citrus by Xanthomonas citri. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are smallest living cells known and can survive without oxygen, that is facultative anaerobic. Many mycoplasma are pathogenic in animals and plants. Diagram of a dividing bacterium shows cell wall, cell membrane, and DNA. Fission. Kingdom Protista, divided into chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoan. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protist to one biologist may be a plant to the other. Chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, the protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. This is the most controversial kingdom. Chrysophytes, diatoms, are chief producers of oceans and golden algae, desmids. This group includes diatoms and golden algae, that is desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, like plankton. Most of them Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells which fit together as in soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups, insulation and soundproofing, etc. Diatoms are chief producers in the oceans. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear in yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has stiff cellulosic plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates, that is, example goniolax, undergo such rapid multiplication that they make the sea appear red, called as red tide. Toxins released by such large numbers may even kill other marine animals such as fishes. Noctulina is a dinoflagellate which shows bioluminescence. Diagram of a dinoflagellate shows cellulose plates, transverse flagella, and longitudinal flagella. Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. The pigments of euglenoids are identical to those present in high plants, example euglena. Diagram of euglena shows a long and a short flagella and pellicle. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form a aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies, which bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years under adverse conditions. The spores are dispersed by air currents. Diagram of a slime mold. So, in suitable condition form plasmodium, unsuitable condition form fruiting bodies, bear spores at tips. Spores are possess true walls. Protozoans. Protozoans are divided into amoeboid, flagellated, ciliated, and sporozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, that is false feet, as an amoeba. Marine forms have silica shells on their surface. Some of them, such as Antamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness, example trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity called gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes water laden with food to be stirred into the gullet. Example paramecium. Diagram of paramecium is shown. Row of cilia and gullet. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, that is malarial parasite which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. Heterotrophic organisms. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. Fungi on a moist spread and rotten fruits are often visible. The common mushroom you eat and toadstools are also fungi. White spots seen on a mustard leaves are due to a parasitic fungi, Albugo candida, which cause disease like white rust of crucifer. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust caused by Puccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cynocytic hyphae. Others have septae or cross walls in the hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores or aplanospores, which are non-motile. Zoospores are motile. And sexual reproduction is by oospores, zygospore, ascospores, and basidiospores. Reproduction in fungi can take place by vegetative means by fragmentation, fission, and budding. Asexual reproduction is by spores called conidia, sporangiospores, or aplanospores. Three of them are non-motile, and zoospores are motile. And sexual reproduction by zygospore, oospore, ascospores, and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves three steps: plasmogamy, karyogamy, and meiosis in zygote. Fusion of protoplasm between two motile or non-motile gametes called plasmogamy. An intervening dikaryotic stage or dikaryophase occurs in basidiomycetes and ascomycetes, which shows N+N stage. Fusion of two nuclei called karyogamy takes place after that, and meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid stage, that is 2N. However, in other fungi like ascomycetes and basidiomycetes, an intervening dikaryotic stage N+N, that is 2 nuclei per cell, occurs. This condition is called as dikaryon, and the phase is called dikaryophase. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of mycelium, mode of new spore formation, and fruiting bodies form the basis for the division of kingdom into various classes. Morphology of mycelium, mode of spore formation, and fruiting bodies. Phycomycetes, example rhizopus, bread mold, albugo, parasite on mustard. Members of Phycomycetes are found in aquatic habitats and on decaying wood, on moist and damp places, or as obligate parasites on plants. The mycelium is aseptate and cynocytic. Asexual reproduction takes place by zoospores, that is motile, or by aplanospores, non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar, that is anisogamous or oogamous. Some common examples are mucor, pin mold, rhizopus, the bread mold, albugo, parasitic fungi on mustard, etc. Diagram of mucor or pin mold is given. Ascomycetes or sac fungi. The ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast Saccharomyces. They are saprophytic decomposers, parasitic or coprophilous, that is growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on a special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asces, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarp. Some examples are Aspergillus, Claviceps, Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. Diagram of Aspergillus is shown. Basidiomycetes. Mushrooms, puffballs or bracket fungi. Commonly known forms of Basidiomycetes are mushrooms, bracket fungi or puffballs. They grow in soil, on logs and tree stems, and in living plant bodies as parasites. Example, rust and smuts. The mycelium is branched and septate. Asexual spores are generally not found, but vegetative reproduction by fragmentation is common. Sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. The basidiospore are exogenously produced on the basidium. Basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom, Astylago smut, and Puccinia rust fungi. Diagram of agaricus is shown. Deuteromycetes or imperfect fungi. Commonly known as imperfect fungi because only the asexual or the vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belonged to. It is possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect or sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes or Basidiomycetes. The Deuteromycetes reproduce only by asexual spores, known as conidia. Mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colitotrichum, and Trichoderma. Examples of Phycomycetes: Rhizopus, bread mold; Albugo, parasitic fungi; Mucor, pin mold. Ascomycetes example: Penicillium, yeast, Claviceps, Neurospora, morels, truffles, Aspergillus, etc. Basidiomycetes: Agaricus, Astylago, Puccinia. Deuteromycetes: Alternaria, Colitotrichum, and Trichoderma. Kingdom Plantae divided into algae, bryophyta, pteridophyta, gymnosperm, and angiosperm. Kingdom Plantae includes all eukaryotic chlorophyll-containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cascata is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants has two distinct phases, diploid sporophytic and haploid gametophytic, that alternate with each other. The lengths of haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomenon is called alternation of generation. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Viruses, viroids, prions, and lichens. In the five kingdom classification of Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroids, and prions. Viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Virus means venom or poisonous fluid. M.W. Bejernik named the new pathogen as virus. D.J. Vaishnavi in 1892 recognized certain microbes as causal organisms of mosaic disease of tobacco. These were found to be smaller than bacteria because they pass through bacteria-proof filters. M.W. Bejernik in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and called the fluid as contagium vivum fluidum, that is, infectious living fluid, called virus. W. M. Stanley in 1935 showed that viruses could be crystallized, and crystals consist largely of proteins. They are inert outside the specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, that is, viruses that infect the bacteria, are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by viruses. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vent clearing, warping, and stunted growth. Diagram of TMV, tobacco mosaic virus. Single-stranded RNA, capsid made of capsomeres, a protein coat, helical structure. And diagram of bacteriophage, double-stranded DNA, infect bacteria. It's polyhedral and differentiated into head, collar, sheath, and tail fibers. Viroids. In 1971, T. O. Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of viroid was of low molecular weight, prions, discovered by Krusiner. In modern medicine, certain infections, neurological diseases were found to be transmitted by an agent consisted of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant, CR-Jacob disease, CGD, in humans. Lichens. Lichens are symbiotic associations, that is, mutually useful associations between algae and fungi. Algal component is known as phycobiont and fungal component is known as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. Lichens are very good pollution indicators. They do not grow in polluted areas. They are highly sensitive to sulfur oxides. Summary. Biological classification of plants and animals was first proposed by Aristotle on basis of simple morphological characters. Linnaeus classified all living organisms into two kingdoms, Plantae and Animalia. Whittaker proposed five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista includes all single-celled eukaryotes such as chrysophytes, dinoflagellates, nucleonoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce by both asexually and sexually. Flagella of protists show 9+2 arrangement of microtubules and made of tubulin protein. Members of kingdom fungi show a great diversity in structures and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are 4 classes under kingdom fungi. The plant includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pterophytes, gymnosperms, and angiosperms are included in this group. The life cycle of plants exhibit alternation of generations, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in kingdom Animalia. Mode of nutrition of these organisms is holozoic. They reproduce mostly by sexual mode. Acellular organisms like viruses and viroids, the lichens, are not included in 5 kingdom.